This is the team from EPFL, Switzerland. Okay, take it away.
Hi everyone, thanks for being here. We're honored to present to you EPFL's 2019 iGEM project entitled ViTEST. Now, all of this started about a year ago when one of our teammates met his cousin, who's a wine grower in the area of Champagne in France. He told us about this disease called flavescence dorée that was plaguing Europe, especially France at the time. And he didn't want his field to get contaminated as well. So he wanted a quick diagnostic tool to detect this disease because it took over 2 months for results to come back in France. This disease, flavescence dorée, is a highly infectious disease with currently no cure available. It's also a quarantine disease under European regulation. It's transmitted by a vector called Cephalodeus tetanus, and it's present in over half of all European wine-growing countries. It first appeared in 1958, but it spread very quickly, and now it's spreading the disease, flavescence dorée, even quicker, with new diseases appearing every year, new incidents appearing every year. This has huge economical impact for Europe. For instance, Italy spent over 35 million euros last year combating flavescence dorée. And it has also a huge ecological impact, as most of the pesticides that are used to fight this disease rarely reach their target species. There's a problem, however, because there's another disease of the grapevine called bois noir, which shares the exact same phenotypes as flavescence dorée. Bois noir is a very non-contagious disease, but they both have no cure, and they share the exact same symptoms. For instance, the leaves curl up and shrivel, and there's giant patches of red on the leaves that you can see clearly. This leads to massive crop loss. Now, could you tell me which disease is infecting this leaf right now? Well, it's actually a half from 2 leaves. One infected with bois noir, the other from flavescence dorée. It's clear, indistinguishable using the naked eye. And a quick diagnostic tool is necessary because you cannot treat bois noir with the same heavy measures that we use to treat flavescence dorée. The vectors for the 2 diseases are quite different. The vector for flavescence dorée, this deadly disease, it transmits it from one grapevine to the next. However, for bois noir, it cannot transmit it from one grapevine to the next. It can only create new incidents of bois noir. So flavescence dorée spreads much more quickly than bois noir and is highly infectious, very dangerous. So we wanted to interact with our local wine growers in the region. One of them, shown here, he's at the epicenter of the flavescence dorée infestations in Switzerland. He told us about these symptoms and his own ignorance and the problems he encountered. And he put us in contact with the regional phytosanitary police. They handle all the sampling and labeling of the infected vines, which are then later sent to the Federal Institute. This was very important to us because he told us about the exact diagnostic method that's used today. Basically, the wine grower goes into his field and looks at the symptomatic plants and identifies them. He then calls the regional phytosanitary police. They then take samples of the leaves, label them, and send them to the Federal Institute for Agriculture, and then they identify the strains and the species. This whole process takes about 2 to 5 weeks to get results back. We were a bit shocked because we thought, well, since it takes 2 months in France, maybe it's just their slow bureaucracy. But in Switzerland, it still took about 2 weeks to a month, so this could not happen anymore. We wanted to eliminate the middlemen that were causing this giant bottleneck and have the wine grower do a diagnostic test for his vines directly on the field, also limiting the use of pesticides and the burning of infected vines that was necessary. And to do this, we have to create a solution that's easy to use, fast, field deployable for the wine grower. But before we show you our final product, here's Theo to tell you how we went about creating it.
We started off our project with one goal, which was to make a diagnostic system for flavescence dorée that would be faster than any current system. To that end, we chose to make our test field deployable. In order to identify flavescence dorée, we chose to detect a DNA sequence specific to this disease. To do so, we start off by extracting DNA from an infected plant, and then, since the concentration we get back is too low for immediate detection, we amplify it. To turn this amplified DNA into a detectable signal, we had it bind to a gene regulator called toll switch, which will then activate a gene creating a color. These last two steps require both transcription and translation, which is done in the cell-free expression system. We will now go into more details on every part of our methods. So we start off by extracting DNA. Conventional DNA extraction methods for plant tissue use chemical cell lysis and take about 3 hours. These methods, however, are lab-bound as they use regulated chemicals, laboratory equipment, and require special training. So we started looking for alternatives that could be taken out in the field and discovered the microneedle patch technology. Microneedle patches are a small 1 centimeter by 1 centimeter patch made of polyvinyl alcohol that have hundreds of micro-sized needles. We contacted Professor Chen Gu from UCLA, one of the inventors of this technology, to acquire the molds necessary to manufacture the patches ourselves. The patch you see on screen here is actually a patch that we made in our lab. But now, how does this work? Well, all we need to do is to apply that patch firmly onto a leaf. The microneedles will puncture the cells, releasing the DNA. Thanks to the absorption and absorption properties of the polyvinyl alcohol, the DNA will stick on the outside of the needles, which means we can get it back by simply running a elution buffer over them. The main advantages of this technology is that it requires no chemicals, that the DNA we get back is amplification ready, that it is extremely cheap to manufacture, and that it takes only 1 minute to perform. We successfully showed that we could extract DNA from blood tissue using our own microneedle patch. Furthermore, we showed that that DNA could directly be used for an amplification step. Okay, so now that we've extracted the DNA, we need to amplify it. However, a standard PCR method would not be applicable in our case, as it requires a thermocycler, which is lab-bound. So we started looking at isothermal DNA amplification methods. We started on loop-mediated amplification, or lab for short, which has already been shown to work in the field for grapevine diagnostics. By that point, we were pretty confident with the general outline of our project. So we could hack the federal body for plant disease detection, to see how we could turn our iGEM project into a viable product. We told them that we were planning on detecting one data sequence specific to Giant Jamboree, but they quickly told us that for a test to become official, it would need both a positive control and to be able to detect simultaneously both Giant Jamboree and the other disease, which is similar, called Bois Noir. That, however, meant that we had to not only detect one sequence, but 3. One for Giant Jamboree, one for Bois Noir, and one for the control, which was supposed to be a chloroplast gene. That meant multiplexing our reaction, and then we're not allowed to do that. We had to go back and look for another method that was called recombinase polymerase amplification. This method is very fast, taking only 20 minutes at 37 degrees. Furthermore, it comes in the format of a lyophilized reaction pellet, which is easy to take outside the lab. Now, if you remember, we want to use that DNA to activate a gene regulator, for which we need single-stranded RNA. To that end, we add a T7 promoter to our amplicon. We've been able to show that we can multiplex our RK reactions using our 3 sequences. Furthermore, we achieve a limit of detection of 10 copies per microliter for Giant Jamboree sequence in microneedle extract, which is equivalent to 1 to the minus 17 molar. I'll now let Laura explain how we can detect this DNA.
Thank you. So, in order to be able to detect the DNA, we wanted to generate a signal. And to do so, we needed an expression system. So, you all know about bacteria, as they're commonly used in the lab. But are you familiar with cell-free system? It contains all the cell machinery needed for transcription and translation of proteins. So, dealing with bacteria is quite time-consuming and complex, as you need to transform, select, and culture them. Moreover, they are subjected to strict lab safety regulations, which prevent us from taking bacteria out of the lab and use them on the field. This is why we chose to work with a cell-free system. It is quick. Your experiments are done under 2 hours. It's easy to use, as you only need to mix all the components, add the DNA, and incubate at 37 degrees. But most importantly, it is field deployable. Since there are no living organisms, we can actually take the cell-free system out of the lab to have a test that is usable on the field. Now, the main problem we have with selfie systems is that the commercially available ones, such as Pure Express, are quite expensive. So to tackle this problem, we decided to work with OnePodPure, a homemade selfie system which was developed in the lab hosting us. So it contains 36 purified proteins, ribosomes, and energy solution. And to create these proteins, it is quite simple. We have to do first one co-culture of all the bacteria expressing these proteins, followed by a single purification. And this leads to a selfie system which is 14 times cheaper than Pure Express. So to show that our system actually works, we expressed SFGFP into OnePodPure, shown in red, and compared with the expression of GFP into Pure Express. And we measured the fluorescence readout into a plate reader. So not only this graph shows that OnePodPure expresses successfully GFP, but we can also see that it has actually a higher expression for GFP than Pure Express. As you all know, iGEM is all about creating and building a community. And we wanted to be sure that the future iGEM teams will be able to create their own OnePodPure. To do so, we characterized all the 36 proteins into the registry. We created a protocol which is available online, and to push this even further, we created a video tutorial which contains all kind of tips and tricks to help the future team create their own OnePod. Okay, so as Theo said, we amplified the DNA and added a T7 promoter to it to express RNA. This DNA will activate a toll switch, which will then activate a gene, which will create a signal. So how toll switches work? A toll switch is a strand of RNA that contains four main parts: the detection region, the ribosomal binding site, the start codon, as well as the protein coding sequence. As you can see, it is shaped in a hairpin structure, preventing the ribosome from binding to its binding site and therefore expressing the proteins. Now, in order to activate the toll switch, we need a trigger RNA. So in our case, we create the trigger RNA from the amplified DNA. Since they have a T7 promoter added during the amplification, once we add these amplicons into our OnePodPure, they will be transformed into RNA, which is our trigger RNA. Now, this will bind to the toll and linearize it. Once the toll is linearized, the ribosome will have access to its binding site and will be able to express the protein. For the purpose of our project, we had to design three tolls: one for Quinoa, one for flavescence dorée, and one for the control. And we used GFP to test if they work. So on this graph, we can see the activation of the toehold. In blue, we have the toehold with the trigger, and we can observe the expression of GFP. In red, we have the toehold without the trigger, and no GFP is expressed. Okay, so fluorescence is great, but it's actually difficult to observe it out of the lab. And since we wanted a user-friendly test, we decided to have a colorimetric signal in the end. So we used CDO, which is an enzyme that reacts with catechol and creates a yellow color compound. So we incubated CDO and catechol into OnePodPure, and we observed that after 1 hour of incubation, we have a change of color, from transparent to yellow. So to sum up our methods, we start by extracting the DNA with microneedle patches. Then we amplify it by RPA, where we add a T7 promoter to it. These amplicons are added into our selfie system, OnePodPure, where they are transcribed into RNA. This RNA will activate the toll switch, which will express CDO and create a colorimetric signal. Now, Leo will talk to you more about how human practices shaped our project.
Making sure that human practices was integrated in our project is a huge part of our summit. We want to make sure that we involve every stakeholder in the current diagnostic method to make sure that we build the most efficient and easy to use product possible. And we interacted with local wine growers that were suffering from flavescence dorée infestation. Now, they told us about these two diseases and how they share the same phenotype, how it affected them, and they gave us useful feedback on our system and how we could even simplify it more for them. We interacted with the phytosanitary police, the regional body that handles sampling of the infected lines, and that gave us all the necessary details about the current diagnostic method and how we could use the infected samples that they labeled for us. Finally, we also dealt with the Federal Institute for Agriculture, which gave us all the biology of phytoplasma and the technical guidance to make an official test that we could actually use in the field. And that shaped our project even more. Now, we integrated all this information and all of our methods into one hardware system. But since you're all here today, and since you can find this on our wiki in more detail, that's the live demonstration. So, here we have our microneedle patch that we talked with at the beginning. It works in the same way. All you have to do is take your leaf, you place it on it, and you stamp firmly like that. There you go. Now it has your plant DNA as well as the phytoplasma DNA. Put it into this little capsule here. And then you use the syringe, which has the elution buffer. You connect it to the microneedle patch. There, pretty simple. Now that you have your DNA, you want to amplify it using your RPA. So, you take your RPA pellet, which is in this tube, you loop the DNA into it, and you let that react for about 20 minutes in your portable heat. Once that's done, you open that again. You take your dropper, you want to make sure you use no pipettes ever. You take the RPA product, and you place it on these 3 paper strips that contain the 1 puff pure cell-free system, which is freeze-dried onto this. You drop 1 drop onto each of these paper strips. And again, you let this react for an hour in your 37 degrees in your portable heat gun. And that's it. Once you're done with that, you'll get a colorimetric signal. It'll produce kind of like a pregnancy test. It'll give you a color here. Now, one of them, this one on the left, will always be activated because that's your positive control. And here on the right, it tells you we have flavescence dorée. Now, also to mention, this has our 1-pot system as well as the toehold for the 2 diseases and for the control. That's why here, flavescence dorée was activated, but 1 Noir's toehold was not. So this leaf is infected with flavescence dorée. And that's it. That's all. Simple to use. Now don't worry, this leaf doesn't actually have flavescence dorée. We showed this to our local wine grower, and he said this test was great, incredibly easy to use. You don't need any special training or lab equipment. And he was so thrilled to hear about it that he said he can envision using it in the morning, then in the evening, getting your results back, and then just tearing out the infected vine and using no pesticides. And now he can smile again. Now, this was just a proof of concept for 2 grapevine diseases. However, the bacteria causing the diseases, called phytoplasma, they affect a number of crops around the world, some of which you encounter in your everyday lives, such as potatoes, rice, bananas, and tomatoes, are all affected by phytoplasma. Now, there's also estimates that about $100 million have been lost in Germany and Italy in apple production due to phytoplasma, and over 10% of the lemon production in Iran has been lost, also due to phytoplasma. We believe that with our easy-to-use test, we can save all these crops from phytoplasma, but maybe not from aliens. We'd like to thank our supervisor and all our TAs, which are sitting right here in the front row.